everybody. Well, thank you for coming. It's a great pleasure for me to introduce today our guest, Simon Verhulst. Simon is a professor at the University of Groningen. Thank you. And he's a leading researcher, especially in the field of aging, where he has made very influential contributions. And he has published more than 180 articles in top journals, and he has received more than 16 citations. And besides that, he has time to supervise more than 15 PhDs and dozen of master students. So I think we're going to enjoy very much the talk today. And yeah, the talk, please.
Thank you, David, and thanks also very much for the invitation. It's really nice to be here. I've been promising to come here for decades, so I'm glad that thanks to David, it's finally come true. And I've really enjoyed the visit to the field site where we were yesterday, and it's probably the best way to get me to make plans to come back as soon as I can. Whether I want it or not, we'll find out, but certainly I'll be interested. So just briefly before I start, I want to say something very general about my sort of research and what I do. Basically, I'm interested in the things that are in this schematic. So we should actually start here because it sort of starts with trying to understand individual variation in life history decisions. There can all be kinds of different things. And I'm interested in how that is influenced and does influence variation in individual state. And both things I'm interested in how they affect fitness. So I very much focus on traits of which it is somehow established, preferably in the context of our own research, that there are clear fitness consequences of variation in that trait. With respect to fitness, as David already mentioned in a sense, one way to look at fitness is to see how it changes with age within individuals, so aging or senescence. And senescence is sort of defined in the context of evolutionary biology as a decline in fitness with age. And so this is shown here for one fitness component for great tit females, where you see that there's a strong decrease in survival rate with age. And you can ask questions about that, which we will. So the first thing I want to make as a sort of a very general thing is that what we observe in terms of senescence, either with respect to reproduction or survival, and please, I may be talking too fast. What do you think? Yeah, okay. So that senescence is in fact sort of an emergent property. So what that means is the following. That fitness components are actually determined by multiple traits that likely interact, that together give an emerging effect on changes with age in either survival or reproduction. So to give an example, here, work I should have published years ago, is showing that on the x-axis age and on the y-axis the daily energy expenditure measured with doubly labeled water of graded females rearing young, or actually females and males, and they were, this was measured at the peak of their, as the working hardest to feed their young, and what you can see that there's a clear decline in how hard they work. So this will be one aspect determining how well they can provide their food to their offspring. So, but of course, the energy for provisioning young is the product, if you like, of the energy spent on gathering food and how efficient you are at gathering food when you are foraging. So suppose, with the starting point that I've just shown you for the decrease in effort with age, suppose that then there's a slight increase in efficiency with age, then this would be sort of the provisioning and presumably also the reproductive success with age. So a slight decline with age. Alternatively, it can be that given the same decrease in how hard to work as they get older, but a steeper increase in efficiency, it may be that actually you find that over much of their success improves. And so this is just a call to sort of think about reproductive success and other aspects of aging as being emergent properties, and it introduces a necessity to think about the mechanism sort of arising, the driving sort of the variation that you see, or the trait that you see. Okay, so we know that fitness declines with age. It's a general observation. In many species, it first increases with age, but we're ignoring it for now. It's another study, other kind of study. And so the question we can ask, of course, is what causes senescence? That's sort of a fundamental question. An enigma. So also in humans, so the root cause being the new fashionable theories coming up every couple of years, but we really still don't know. And first thing to establish is that age in itself does not cause changes in fitness. It's changes in phenotype with age that cause the change in fitness. So this goes a little bit back also to saying that senescence is an emergent property. So there's two questions we can ask about this, if you like. The first one is, how does the phenotype change with age? And secondly, how does the change in the phenotype affect fitness, or is that driving changes in fitness? So the general approach we take in trying to study this is that we do environmental manipulations, you see here, that affect fitness. And once we've reached that stage, that's just the starting point, if you like, because at that point, we can start to study aspects of the phenotype and see at the same time how they are affected by our manipulations, and how and to what extent they explain fitness consequences of the manipulations we do. So this is what we started with. We started with manipulating reproductive effort of birds. And so basically this goes like this. It means you have two broods of, say, 4 young each, and we move chicks around to create a smaller brood size of -2 and a larger brood size of +2. So we have reduced broods and enlarged broods. In our design, for statistical reasons, we don't actually have a control.
I should talk like this, is that it?
So we changed that, but that's not relevant for this talk. Now, in a sense, I would not recommend anyone to do this now. And the reason is sort of explained in this meta-analysis done by Eduardo Santos and Shinichi Nakagawa, published some time ago, who analyzed the large number of studies that have been done in birds where brood size was manipulated. And what they found is that in females, actually, brood reduction in either sex had no effect on survival. There was a weak effect of enlarging brood size in males, but all in all, you could say the evidence is pretty weak. And to me, when this came out, I actually thought this put the field of avian evolutionary ecology in a crisis, because thinking that there are sort of costs to what individuals do is sort of really central in our thinking. It's a crisis that seems to have gone unnoticed, or maybe everybody chose not to mention it, because I never encountered anything in writing or speaking where people say, we've got a serious problem. But I think luckily our design was slightly different from the design used in these studies. Namely, we did not stop by manipulating brood size in a single year, but we would continue to do that more years. And what I mean here is not that we sort of did the brood size manipulation multiple years, but if an individual was assigned to rearing either reduced or enlarged broods, it would be rearing reduced and enlarged broods for the rest of its life. And this being done in jackdaws that have quite a long lifespan, that asked for serious commitment on our part. So it's luckily I don't have a lot of fantasy and wouldn't want to think of anything else to do either. So we just continued and continued. So we maintained these manipulations for life, and actually we started in 2005 and we're still doing it. And this is what we find with respect to survival. So what you see here on the x-axis is the number of years in treatment, so the number of years they've been manipulated, and on the y-axis is the annual local survival. in a moment I'll show you a more sophisticated analysis that takes into account observation probability. But these are the raw data. And what you can see first is that in the first year, there's no effect at all. So that's very similar to the results of the meta-analysis, namely that manipulating brood size in 1 year doesn't affect survival very much. However, what you can see is that in later years, over the time that we continue the manipulation, the open dots are the parents with reduced broods, that their survival is better. And so when we continue the manipulation, we find a clear fitness cost of rearing and enlarged broods in terms of reduced survival. Eyeballing this, you can also see that the difference in survival seems to be time. So it was very non-existent the first year, and then there's a small difference, and you can see in the last years there's a really large difference. And we were interested in modeling this in more detail, because the question we were interested in is the following. So when you have an effect on lifespan, it's sort of possible that there's sort of a fixed effect independent of age. Here there's on the lifespan, or it can be that the effect on lifespan systematically changed with age, and in particular, there was the possibility that sort of seems to be there in this graph, that really the rate of senescence, actuarial senescence, so that's the increase in mortality with age, is different between the categories. So to model this, we fitted the data to the Gompert's hazard function, which looks like this. And I'll just briefly explain what it's like, in part because I hope that other people will follow this example and do this also. So it's a simple equation. It's A times e to the power b times t, and t here is time. So that would be age or number of years in treatment in the context of our experiment. So what you can see is that this b sort of is the exponential part, and that determines the effect of age. And a here is a sort of the initial mortality rate. So if you set t to 0, then this reduces to 1. So the mortality rate is then a. So what happens when you change either a or b is shown here. So on the left is the sort of the proportion of the cohort surviving as a function of time or age. And in this graph, you see how mortality rate increases with age. And notice that this is the log of mortality. So this continuous line is sort of the starting point, and the two lines here shows what happens if we make a change in either A or B. So we change A or B down. So if we change A, reduce A, then what happens is, you see in this curve, and the curve just sort of shifts to the right. But if you have a change in B, then the decline becomes more gradual. So that means the increase of mortality rate with age is more slow. So here you see that in terms of mortality rate. So this is the starting point. Then there's a parallel line here when there's a change in the initial mortality rate. But this is a less steep line, and that is when there is a change in the rate at which mortality increases with age. So we fitted the data from all those years to this equation. And so that's done using a dedicated package, our package that also takes allows to take the capture probability into account, which you fit separately. And so it says B0 here, that's Basta speak for A. So these are the posterior distributions. And you can see there's no effect at all of the manipulation of the initial mortality rate. However, there's a clear effect on this B1, which here is this parameter B, meaning that there's a higher aging rate in birds rearing enlarged broods. And that is shown here. So just to remind you again that this is a log axis. So actually the increase is much steeper. So we have linear minds which are not really suitable to interpret log axis. And this is the number of years in experiment. This is just the graphic display of showing how mortality rate increases faster in birds rearing enlarged broods compared to birds rearing reduced broods. So that's quite, so maybe that's the crisis in avian ecology, evolutionary ecology averted, again without anybody noticing. But at least to me it was sort of satisfactory in that sense. But of course, this now is only the starting point, because we now have sort of created a suitable context in which we can start to understand, try to unravel what happens on the phenotypic level that explains this variation in this experimental effect. And here's another problem, namely that phenotypes are actually quite complex. They're multidimensional. And it's well known that traits change with age in different ways, and we label that as mosaic aging. So it's one thing to say, okay, so we've had this experimental effect, now what happens on a phenotypic level, but then lots of traits change with age in different ways. So this is illustrated here for a long-term study by Michael Briga and others. Blanca actually was very important in this study as well. And what Michael here is sort of summarized for different traits: body mass, basal metabolic rate, standard metabolic rate, hematocrit, how they changed with age. And so you see that body mass sort of increases and then decreases, and this trajectory is faster in birds that have a short lifespan compared to a long lifespan. Basal metabolic rate decreases with age, but is independent of lifespan, both in terms of rate and level. Interestingly enough, standard metabolic rate, measured under exactly the same circumstances in the same individuals at the same time, except that there the temperature is lower than the thermoneutral zone, means they expend extra energy, actually increases with age, again is unrelated to lifespan, and so this is more of the same. The B color is actually quite an interesting, very different pattern, but that's another story. So this is a thing. So when considering what traits to measure to understand or unravel the causes underlying the experimental effect, we have this to deal with, this question that there's different traits varying in different ways. And what we hypothesized is that possibly the fitness consequences of changes in trait value, that they are likely to differ between traits. So you can easily imagine this: if your gray turns hair, if your hair turns gray, this was unlikely to affect your fitness very much. But at the same time, you can think of lots of physiological traits that if we would change to the same extent, say your blood glucose level or whatever, or your body mass index, then they would actually affect fitness quite a lot. So in that context, it seems likely that if the change in a trait is more important for fitness than the change in another trait, then that change that is more important is likely to sort of be regulated to change less. So that's the basic idea. So we have that in a graphical version as well. So imagine this is the trait value on the x-axis, and let's suppose for a moment that this is the optimal value for both trait B and trait A. And what is on the y-axis here is sort of the marginal fitness costs of having a deviation in that trait value from the optimum. Now suppose birds or individuals, it can be any species, are at the optimum, but there is some kind of perturbation in the environment causing the traits to deviate from the optimum. And at that point, of course, you expect a response on the physiological or behavioral level to bring traits back closer to the optimum. And here's the thing, that the obvious expectation is that they will be brought back to the level where the marginal fitness cost of the deviation is the same. Because at that point, it's no use investing more in one than the other. But that means, of course, that the trait with the higher fitness cost of the perturbation will be brought closer to the optimum than the trait that is less costly, say the gray hairs. Yeah? So that is an interesting thing. That's the way we think about, and we tested that in the jackdaws again using the brain science manipulation. So not in the context of aging, but I think that conceptually it's the same. And so we have these birds growing up in either reduced or in large broods, and we measured lots of things on the physiological level and also some not physiological level in these individuals. And they then fledged, and we could see in the years that came after that who of those fledglings would survive and who didn't. So who returned to breed or who did not. And we could relate the things that we've measured to the probability of surviving. And so the prediction here is that the traits that responded very little to the brood size manipulation, so they were well protected in the physiology of the animals, that those would then be the traits that would predict whether they would survive, while the traits that responded strongest to the brood size manipulation would not be predictive of survival. And that's indeed what we find. So you see here on the x-axis the effect on the brood size on the different traits. So most of them were oxidative stress related. And on the y-axis is the association between that trait and the probability of surviving to come to breed. And there's a significant negative association between the two traits. So an interesting but perhaps frustrating consequence of this finding is that if you do experiments and on some things you find strong effects and others you don't, you may be inclined to follow up the strong effects, but it may be the things that on which there's no effect are actually the most interesting ones. And then you have to defend, no, there's no effect here. This is what I need to study. And so that may be a difficult thing to defend, but I think nevertheless that's the thing that would be sensible to do. So basically, the fitness consequences of changes in trait value are likely to differ between traits, and we think this may be key to understanding mosaic aging, if you like, so variation in aging rates between traits. Okay, so we've just made things more difficult. I can make it more difficult still, because we still have the question, how do we best measure state or condition, or whatever you prefer to call it, in the context of these experiments? Another approach is to sort of measure as much as possible and use structural equation modeling, as we've done here. And it's quite elegant in a sense, because what you do is you derive an estimate of condition of individuals using all this information on all these traits. So we integrate it in one sort of variable, in one estimate, and how you integrate them, so what weight you give them and whether or not there are interactions or not, you let depend on the extent to which this condition rate, these traits will affect survival. So it's quite elegant in a sense. Wouldn't recommend it though. You rarely sort of have the consistent data time, so having something that you then can transfer to a new data set on the same species or so, even that is sort of difficult. But it's a very interesting analysis that tells you much about the relation between the different things you measured and how they contribute to variation in fitness or survival or whatever you're looking at for. Okay, so what we actually need is one trait to rule them all, and one trait to find them, and one trait to bring them all, and in our research bind them. That's what we're really looking for. So that's what I've been trying to do, and this is what initially turned me to look at telomeres. So telomeres are the ends of linear chromosomes, and they have this sequence TTA-GGGG that is sort of repeated thousands of times. And it's evolutionary, very well conserved. All vertebrates have it. Insects have sort of slight variants of it, with one base pair being slightly different. And so before coming back to the experiment, I want to say a few general things about telomeres, which are probably not too new, but nevertheless. First, using the pedigree of the jackdaws, we estimated the additive genetic effects, the additive genetic variation in telomere length, and we find that when we have 4-day-old chicks, then about 74% of the individual variation in telomere length estimated with these animal models can be attributed to additive genetic effects. When we sample them again just before they fledge, the result is the same. And this is because the changes in telomere length over the nestling period, which I'll show later, I think, can be quite important. They are really small relative to the large range of individual differences. So there's about 3,000 base pair difference between individuals with the shortest and the longest telomeres, while the change within an individual over this period is about 250 base pairs, so of that order. Further, what's a general thing is that telomere length declines with age. So this is for a thing we did in humans. Typically, this decline is faster early in life, and then gradually becomes more shallow. But the reason this is all interesting, and that's the reason actually that I got into telomeres in the first place, is that telomere length predicts survival. So these are human data that we did on 3 cohorts in the States that were really well studied for a longer period. And what you see is the hazard ratio on the y-axis, and it's normally plotted on a log scale, but we plotted it here on a linear scale because a log scale sort of muffles how big the effect really is. And what you can see there is that if you have short telomeres, then say at age 80, your mortality rate is about double as what it is when you have long telomeres. So these are sort of the ends of the distribution as we see them. I think it's, oh, that's there. So it's minus 1.5 standard deviation, minus 1.5 kb to plus 1.5 kb. So that's actually 2 standard deviations. Okay. So this makes telomeres interesting. So there's other reasons to think they're interesting. So for example, this is work we did on purple-crowned fairy wrens. The work is driven by Anna Peters at Monash University in Melbourne, we collaborate with. And what you see here is the relative telomere length of nestlings when they're 7 days old. And what you see on the y-axis here is the contribution to population growth, so their fitness. So this is really sort of their fitness. It's mostly driven by lifespan. And I think this is an amazing picture, an amazing result, just because of the idea that you take a few drops of blood of a 7-day-old nestling, and that is then a significant predictor of how long it will live. I find that still astounding. And so it's never very precise, and the predictions are never sort of really accurate. You couldn't use it for individual humans or so. At least we haven't tried. But that there are these patterns at all still sort of blows me away to think about it. That you can measure this one trait, and the birds, they can live for years, huh? They can live 10 years or so, but still it's this predictive. Okay. So back to the jackdaws, where we did the brood size manipulation. And now, of course, we want to know whether the accelerated aging we saw we gave them enlarged broods, whether we see that also in the telomeres. So first, also in the jackdaws, we find that individuals with long telomeres have better survival in adults. So this is plotted in a way that reflects how we analyze it, which I understand is not so sympathetic, but there you are. So this is the between individual slope of telomere length, and this is the within individual slope, and the fact that this slope is much steeper is evidence for what we call selective disappearance. It means that individuals with short telomeres disappear from the population, and that's why the average telomere length sort of doesn't change so much. So the prediction that we now have is that for the experiment is that birds with rearing reduced broods, that they will lose their telomeres at this slower rate than birds rearing large broods. That's the prediction we have, given that we have this experimental effect on aging, and secondly, we know that there's an association between telomere length and survival. Nevertheless, this is what we find. So there's no association at all. There's no experimental effect at all on the rate of telomere loss. And this is actually beginning to fit what we know about telomeres very well. So the telomere length seems to be set quite early in life and does not seem to be so open there's little individual variation in shortening rate thereafter, at least in jackdaws and humans. You can see I'm a bit cautious. There's some results in the literature and of work in other species where that make me doubt and I think that individual variation in telomere dynamics does seem to be informative sometimes, but I think it's not nailed down yet, so I'm not sure. So about this sort of persistency, so this is the telomere length of nestlings, and plotted against the telomere length when those birds came back as adult. And this dotted line is where they would lie on if there was no change in the meantime. You can see there's a clear change in telomere length. But you can see there's a very strong correlation between the telomere length they had very early in life and the telomere length they had when they came back as adults. And mind you, these are there are several years in between. They come back to breed on average when they're 3 years old, and it's quite variable. So that will explain some of this variation. And of course, there's a lot of telomere shortening occurring very early in life, and it presumably sort of carries on also after we take the last sample, which is sort of on an arbitrary day. It's not arbitrary at all, in the sense that if we do it later, they're likely to have gone. So it's very practical, but it's arbitrary from their perspective. If we look at the adults, if we take the first telomere length measurement in adulthood and we plot that against the last telomere length measurement in adulthood we have of those same individuals, you can see there's incredibly tight correlation. And the sort of these different lines, which you can probably not make out, they show that interval between the first and the last measurement is longer. The line will sort of become slightly lower, but they all run parallel to each other. So this is jackdaws. In humans, we find the same. So this is data from humans, adults that were sampled with 12 years between baseline and follow-up. And you can see there's an incredibly tight correlation between their telomere length in the two samples. And the R square is 0.94. And actually, so I actually haven't done the exercise, but I know that from these data that the measurement precision is just very high. And the deviation between 1 and 0.94, I think most of it, if not all, can be explained by measurement error. So it's really almost perfect. So the take-home message, but remind you, it's a slightly personal view, is that adult telomere length provides information on phenotypic state. Remember these associations between telomere length and survival, but not on telomere dynamics, so not on changes in the phenotype with age. That's our view on the moment. And so, in other words, contrary to popular belief, telomere length is not a marker of senescence. And I'm going to repeat that. Telomere length is not a marker of senescence. Yeah? It's an interesting marker of individual state, but not of senescence. So that's an interesting paradox. So, exit telomeres. But still looking for an aging biomarker. Mind you, it's not completely exit. It's exit telomeres in the context of finding a biomarker. Lots of context in which it's clear that. I need to say this in part because I encouraged David to do some telomere work this morning. So we went to sort of another thing, which is DNA methylation, which is sort of what we are investing our time and money in now, in addition to the telomere work. And basically what that amounts to is if this is DNA, then there are sort of methyl groups that attach to the DNA, and this is a tightly regulated process, it appears. And what makes this interesting is that the degree specific sites are methylated changes with age, and that can be changes either negative, 0, or positive, so that all happens. And they're very robust, and what has been done in a very interesting way is that the methylation information from different sites can be combined in an index of age. So that means that if you know the methylation from enough individuals and you know their age, you can use those data to generate an index of age, of how, so that predicts chronological age. In humans, this can now be done with a precision of about 2 years. So that means that if you give someone a little bit of your blood, they can tell you within 2 years how old you are. People have been doing this sort of in non-model organisms, and we reviewed that recently, and there's lots of general discussion, so if you're interested in this work at all, then this is a paper. There's one of 2-3 papers I would recommend you to read, to sort of just to get into it. And what you can see here is that for these different, where this has been estimated, you can see that on the whole, produce quite strong correlations between the chronological age and their sort of epigenetic age. Of course, for an aging researcher, this is useless. The reason is simply that when we study aging, we usually know their age. So having a marker to know their age is not so useful. I mean, if you don't know their age, what are you doing? So what makes it interesting is that it turns out there's quite a robust result in humans so this is chronological age on the x-axis, and this is sort of epigenetic age on the y-axis. What they find there is that if you have a relatively high epigenetic age for your chronological age, you also have a higher mortality probability. And conversely, if you are relatively young for your chronological age, you have a lower mortality probability. So it really seems to be sort of a measure of biological age, rather than, so if you know epigenetic age and chronological age, then together you can get an estimate of biological age. How to define biological age exactly is another discussion, which is interesting, but not for now. So we've just started investing in this, and this is a new field for us, so I'll be showing you some relatively new results. So just the first two papers we're writing up with sort of descriptive stuff. So what we see first, so this is the two species we do this, and zebra finches and jackdaws. I didn't mention much of the zebra finches, but also in the zebra finches we've done experiments. I showed you Michael Briga's mosaic aging results, but we find sort of experimental effects on aging in that context also. So this is the methylation, the average on a site, or on a chromosome in this case. And so the two points sort of joined by black lines, they are sort of the same individual sampled when there were different ages, so either young or old in this context. And the different lines from bottom to the top, they are different chromosomes. And so what you can see is that, firstly, that there is very consistent differences between chromosomes. Some chromosomes are highly methylated in all individuals, and some are sort of very weakly methylated, and this is all the same in the jackdaws. What is slightly less visible, but is nevertheless the case, so is that this methylation overall sort of declines somewhat with age. Of course, this is not so interesting yet when we're looking at an aging index, but it's a little bit interesting. And further, what you see is that the sex chromosomes, they behave differently. So the W chromosome in the zebra finches sort of has higher methylation than any other chromosome. In the jackdaw, it's also relatively high. The Z chromosome is highly methylated in the zebra finch also, and but not in the jackdaw, so it seems. This large variation between chromosomes in methylation is largely determined by the of the chromosome. So if this chromosome is larger, so this is the number of base pairs, then the proportion of CpG sites that is methylated initially decreases, and you can see over a fairly large range it's flat, and here it's a slightly more consistent decrease. And there you can see that the W chromosome is much more methylated, and the Z chromosome also in the zebra finch, and in the jackdaw it's just a W. We were interested in looking at this because of the dosage compensation that happens on sex chromosomes, given that, of course, here the males have 2 Z chromosomes and the female only 1, so there needs to be some level of dosage compensation there. So in humans, actually, in females, completely deactivated. And in birds, it's well known how it works. It's not methylation here because there's no indication that the one male Z chromosome is more methylated there. So the next thing we did is we said, okay, so this is all in a relatively small sample of sort of 10 individuals in both species. And we're sort of looking at the larger set now as we speak. Next, what we wanted to do is, okay, to what extent can we find age-related, to to what extent can we find CPG sites where the methylation is age-related in these individuals? And what is new in the field is that we were doing this longitudinally. So we're really looking at the extent to which methylation changes with age within individuals. And so we calculated the degree of methylation for all sites, for all individuals. And when I say we, you probably know what that means. So the different steps, we selected the sites that were most strongly correlated. Correcting for a statistical artifact generated by variation in mean coverage. And then we did a little bit more selection, and then this is what we get. So we selected about 1,000 sites in both species that were really strongly correlated with age within individuals. The first thing that's worth noting is that in the zebrafinch, there seems to be slightly more sites where methylation is increasing than where it's decreasing. So this is the proportion of increasing and decreasing sites of the age-related sites. Well, in the jackdaw, there's clear that decreasing is much more frequent than increasing. If this is interesting, if this means anything, we don't know yet. We're sort of scratching the surface, exploring new data, seeing what is there, and it's a bit intimidating at first, but then it's just data, so you can play with it as you do. So then we looked at the distribution of these age-related sites over the chromosomes. And there we have an interesting pattern that in both species, the W chromosome in particular seemed to harbor a large number of these age-related sites. So that was a striking thing. And again, of course, to look at correct for how many CPG sites there are on that chromosome. And so what you see here plotted on the x-axis is the number of CPG sites per chromosome. So each dot is a chromosome, and on the y-axis is the number of age-related CPG sites. And so, of course, it is positive. So the dotted line is what you sort of expect by chance if there's no chromosome-to-chromosome variation in the likelihood to harbor these age. And what you can see here is that than you expect by chance. And actually, the Z chromosome seems to harbor a bit fewer. And in the zebrafinch, both sex chromosomes harbor more of these sites. These are two other very small, so microchromosomes, that also seem to have a lot of these sites. So this is where we are. We're now in the process of actually a larger set of individuals. So this is how quickly the field develops on a technological level. We thought we're going to have these 1,000 sites, and then we're going to go back with a larger set of samples and have the methylation measured in just in those sites. But it turned out to have the methylation measured in 20 million sites only cost twice as much as the cost of having it did in 1,000 sites. And so now actually we're going to do everything, whole genome. And so we have a large set of zebrafinch genomes waiting or being analyzed now, and jackdaw genomes on the way. So, of course, all this was done by a lot of people together. And so I'm really grateful to all of them. And some of them are in the room, which is very nice. And I'm happy to answer any questions.
Thank you, Simon, for this interesting talk. So, time for questions. Any questions?
Hello. Thanks, Simon. Very, very interesting, as usual. So I have two questions. One is more on your personal view or your personal opinion. So regarding the telomere shortening lack of root manipulation effect on the jackdaw with age, I was wondering whether then that fits in with your prediction of that is a trait that is canalized, well canalized, and therefore then if we try, so what would a manipulation on the opposite direction, so trying to counteract telomere shortening, then have an effect on fitness?
Yeah,
positive benefit?
So actually, so the question is whether, so according to this scenario that I sketched, you could say, well, so telomere length or dynamics clearly is a predictor of fitness components, so you would expect it to change little in response to any stressors. That is under the assumption that the association between telomere length and dynamics and fitness components is causal. And I actually think it is not. So I think it's, and again, you'll find some of my close colleagues having a different opinion, but I think it's not, and actually from time to time they concede actually that they're wrong. Just to illustrate, this is sort of the telomere dynamics I've shown you already for humans, and we can also look at, so you can wonder, okay, so are people dying because their telomeres are sort of getting really short? The answer is we don't think so, because when we look at patients with a telomere disease, so they're genetic diseases causing telomeres to be really short, we find that their telomere length, the telomere length they have, so it's given by these closed dots here, is rarely, if ever, reached by humans in a natural population. There's some caveats here, and that is, it can always be argued, but this is the average telomere length, and maybe it's the shortest telomeres that cause. The evidence for that is based on cell cultures, and the extent to which this happens in vivo, we don't know. In the bird studies we've done, we find the opposite. There is a new development of telomeres being measured with nanopore technology, and there it's possible to get data on individual telomeres sort of within a sample. So you actually, with one measurement, you get, say, 300, 400, 500, 600 measurements, and sort of there the telomere sequenced all the way up to the subtelomeric region. And so possibly with those data, we can reject the hypothesis that it's the shortest telomeres that cause this effect. But until we do, that remains an open question. So to come back, I think telomeres are informative because they're not important. That's how I think it worworks.
Okay, and then my second question, if I can steal more time.
Up to David.
Yeah, regarding the DNA methylation on the zebrafinch, the patterns that you found, I was wondering whether you have any ideas or you are planning to look on what genes, so what are the coding genes in those chromosomes that are being more methylated?
So these are results I got yesterday morning. So this is the distribution of the age-related CpG sites. So they are very preliminary. As I said, I received this morning. We still need to discuss the details. But I trust the people who are doing this for us. So basically what you see, there's a fair proportion in the promoter, a bit less in the jackdaw, and these are intergenic, so these are sort of informative, and these are just around and need to be skipped. So one option we have, and that is really interesting to consider, I think, is that when we are going to build... So I should go one step back. A thing that I didn't mention at all is that the DNA methylation is interesting, not just because it can be used to estimate biological age, but also because it has a direct functional consequence. If a gene is methylated, in particular if the promoter is methylated, it reduces the expression of the gene. So that means it's possible with this to make sort of functional links also with the index we find. And so the sort of decisions we need to take when building indexes is whether we should sort of select sites in promoters or maybe in the intergenic region specifically, and of course we're probably going to do it all and compare the results so we can say something about that. And so you can imagine that if we have one that is in the intergenic region, that it will be sort of really informative, like the telomeres, while if it's in the promoters, it may be sort of really canalized. So that maybe it's possible that it will be important in predicting fitness, but will not be affected by experiments. So anyway, we're going to explore all that. Anyway, don't forget you saw this. This is really preliminary. So.
More questions?
Thank you, Simon. It's not about your work, it's about the meta-analysis you show at the beginning. The lack of effect of the manipulation on survival could not that be linked to the average lifespan of that species? I mean, wouldn't you expect that manipulation to be very costly for a short-lived species, whereas something like your jackdaws?
I've been looking for a partner to look at this in detail, if you're interested. I think that, so it's sort of, so on of my sort of too long to-do list of things I'd like to do at one point is sort of go deeper into this and see whether we can understand the variation in effects. To be honest, I'm fairly sure that Shinichi saw this and the set, but I don't remember the result. But I imagine there wasn't an effect, or I would have remembered it also, but I'm not sure.
Also, a short question about your canalization study. Those traits that were not very important for fitness, but they change a lot, are those changes that are necessary for, let's say, very quick manipulations of the phenotype? I mean, things that are extremely necessary at the time of growing. Things like a short-term, long-term kind of...
I think there are too few traits that we know too little about to really say. But, but of course, you know, the organism is a very dynamic thing, and there are sort of short-term needs and long-term needs. And I think it's interesting to think about, but not so easy to come up with something straightforward. Also, you can, it's thinking about which traits are sort of useful for this analysis and which are not is also something like, because you can, okay, so what kind of phenotypic plasticity and at what time would there have to be? So I tend to think that sort of, for example, sort of physiological traits, because really sort of they're relatively dynamic, I think are more important for traits that are less plastic in the short term. But we really need more people trying to sort of test this idea further and think about it. I think of course we're the first to do this. We got away easy, but and now it's time to sort of start thinking more critically. Okay, so to what traits do we expect this to apply, and to what do we not? And so also related to Elisa's question. Should they be causally related to the fitness consequences, or can they just be some markers that have no direct effect? And so there's lots of things to think about there. Mm-hmm. I think I'm quite confident about the principle, but actually to address and test it, I think this was okay. I'm reasonably convinced myself.
It's a super result. Yeah.
But it really needs more thought to before it can be broadened.
Yeah. Thank you.
Yeah.
Very nice.
Thank you, Diego. More questions? Come on, don't be shy.
Well, thank you, Simon, for the talk. I mean, it's been very illuminating in some ways, some puzzling in others. I just wanted to encourage you to make a small exercise of speculation, if you feel like it.
I don't worry. I'm happy to say anything.
Yeah. Okay. So, just wondering whether it might be the role of environmental variation on the evolutionary trajectories of aging, of senescence. Sorry. So because, I mean, if you were saying that senescence is a decrease in fitness with age, but fitness is quite environmental dependent, I just wonder what might be the role of environmental variation on senescence.
Well, so the driving thing that seems to be important, certainly on an evolutionary timescale, but also in interesting examples within an individual's lifetime, is the extrinsic mortality rate. So we know that when extrinsic mortality rate in adulthood is high, then fast life histories evolve. When it comes to the evolution of senescence and environmental factors, then that is the first thing that comes to mind. There's other stories to tell for which I don't have time, but I think that some of these, well, I can summarize it. So story I still really need to write up is how ecological circumstances can drive variation in lifespan. So that's related to senescence. So the study I'm referring to is that what we analyzed is that so we know that those seabirds that travel very far to get their food, that they have very long lifespans. And if you look at all the things of their reproductive biology, at least through my glasses, it can be interpreted as all being sort of selected to minimize the amount of food the brood needs per day. And so typically those birds also, they have to be quite old before they start reproduction, and it really seems that they really need to acquire the experience and the proficiency to gather enough food. So they're really sort of at the brink of what can survive and reproduce in that environment. And so my perspective there is really sort of demographic. And the thing is that the only species or populations that can persist in such an environment, where reproduction has to be very slow, are populations that have very long lifespans. So it is a matter that, if you like, the environment will have selected life histories because, say in this case, very slow life histories, because the ecology does not make a fast life history possible. And so that is a completely different ecological level, and on where you see interesting patterns. So you see a very strong association between how far these seabirds travel to get their food and how fast their life history is. So as they go further, the life history is slower. And so that, if you like, is so that next to the extrinsic mortality rate is sort of another sort of an ecological driver. I've been looking for other examples of systems of variation where you see the same thing, and if anyone comes up with something, I'd really like to hear it. So in particular, when there are multiple species on a gradient, as is these seabirds. So a colleague of mine has been studying lark species in different environments along, say, an aridity gradient. And there, the little data that there are sort of also suggests that in these more arid environments, the life histories are very slow. And also there, it's easy to conceive that only a slow life history could persist there. Because only if you can reproduce many times can you replace yourself in your lifetime. While in a more temperate environment where the productivity is much higher, there actually is an option to go fast. So extrinsic mortality rate, habitat productivity, but in a different way. So the one is sort of evolutionary with optimization, and the other is actually through selection through the habitat and the productivity. Another long answer, I'm sorry.
That's very satisfying, thank you.
Hi, I was wondering about the individual consistency in telomere length, and this would go, I think, in the same direction. There's individual consistency in telomeres, there's individual consistency in other physiological traits, So does it mean that the evolution of these traits related to fitness is not so much linked to variation in environmental characteristics?
Well, actually, I think
Because
telomere length is quite unusual in how consistent it is. The repeatability of physiological traits, I think, is not that well known, and as far as we know, it's not that high. So while sort of the repeatability of age-corrected telomere length of adults in jackdaws and humans seems to be pretty close to 1, for physiological traits, it's probably, if it's 0.5, it's quite substantial. And so most of these things, but then there's a whole sort of, there's a whole range of temporal dynamics for different traits. And so that in itself, it's not something that physiologists study. So it's people like us that have to do that. But so some things change very quickly, and others change very slowly, and others never at all. And so and how they differ in the extent to which they predict fitness, I think will be very interesting to study. But at the moment, I don't think I can say anything sensible about it. Maybe after lunch.
So thank you. We don't have more time for questions.
Yeah.
Thank you, Simon. But we can continue talking about this in the kiosk, so we are going up there now. So we hope to see you there. Thank you.